Social touch promotes interfemale communication via activation of parvocellular oxytocin neurons. Oxytocin, the love hormone. Oxytocin is important for maternal behavior and social bonding. Examples of maternal behavior include grooming, licking, and oxytocin also can induce uterine contractions. Oxytocin is produced in the paraventricular nucleus, or the PVN, and the supraoptic nucleus of the hypothalamus, and is released by the posterior pituitary. Activation of oxytocin receptors results in behavioral changes. Oxytocin gives rise to a positive feedback loop. The image in the bottom right corner shows how oxytocin results in a positive feedback loop in 3 different scenarios. On the left, we have a positive loop in the individual. In the middle, we have a positive loop in regards to maternal behavior and bonding. And on the right, we have a positive loop in the next generation of rats and rat pups. Background information. The purpose of this study is to explore the activity of oxytocin neurons during social interactions. Most oxytocin neurons are magnocellular neurons that simultaneously project to the pituitary and forebrain regions involved in social behaviors. This study focuses on parvocellular oxytocin neurons that are activated by somatosensory stimuli and synapse onto magnocellular neurons. These neurons project to either the posterior pituitary, where secretion of oxytocin in the blood causes physiological effects, or to various regions of the forebrain, including the medial prefrontal cortex and hippocampus. Previous studies suggest that female-to-female interactions, unlike male-to-male or male-to-female interactions, are mediated predominantly by somatosensory input. Materials. The materials used in this experiment are listed on the right side of this slide. They include antibodies, eukaryotic cell lines, and female Wistar rats. Ex vivo electrophysiological data was collected using the pClamp10, an Axon instrument. In vivo data was collected using OpenFS GUI software. EthoVision XT 11.5 was used to record behavioral experiments. Methods. Subjects' PVN were bilaterally injected with a recombinant adeno-associated virus. This injection induced the expression of channel rhodopsin 2, a light-sensitive ion channel, when under the control of an oxytocin promoter. Implanted tetrodes paired with an optic fiber were used to record individual neurons in the PVN by identifying their electrophysiological response to blue laser pulses. 90 neurons in 10 female adult rats were recorded. Behavior and ultrasonic vocalizations during open field and free social interactions were monitored. Only virgin female rats were used in these studies. Methods continued. Neuronal activity was recorded when rats were separated by a transparent wall with small holes. This allowed rats to hear, see, and sniff each other, but blocked touch. By removing the wall, free social interaction between the 2 rats was allowed. Air puffs were used as tactile stimulation in isoflurane anesthetized rats to determine if somatosensory stimulation in itself is sufficient enough to increase oxytocin cell activity. This slide shows the setup of the cages during the experiment. On the left, we have open field, where there is only 1 rat in a cage. In the middle, we have chambered social interaction, which is characterized by a wall with small holes separating the 2 rats, as discussed in previous slides. On the right, we have free social interaction, where there are no barriers separating the 2 rats. Below each image, the average firing rate is listed, and there is a bar showing oxytocin spikes. Results. Social interactions activate PV and oxytocin neurons. It is hypothesized that oxytocin neurons were mainly activated by physical contact, since distinction between individual ultrasonic vocalizations between the two rats was not possible. 90.4% of channel rhodopsin 2 expressing neurons were oxytocin positive. 15 of the 90 recorded neurons present in 5 rats were identified as single oxytocin neurons. Air puffs induced a significant increase in spiking activity in 83% of recorded putative magnocellular oxytocin neurons. However, they had little to no effect on activity of non-oxytocin PVN neurons. Results continued. There was little difference between spiking activity of oxytocin neurons when comparing the chambered social interaction and baseline recordings in an open field. Neuronal activity after removing the wall to allow free social interaction showed a significant increase in activity of the same firing neurons. The greatest change in firing rate occurred when one of the rats was crawling on the other. The graph to the right shows the change in firing rate of 5 different social behaviors in respect to the baseline rate. These behaviors are sniffing, chasing, head-to-head, crawling on top, and being crawled on top of. Statistical significance is represented by a star. This graph shows the average firing rate of the 15 oxytocin neurons in the 5 virgin female rats. The index of dispersion and the average pairwise Pearson's correlation of spiking activity are also included. Discussion. Parvocellular oxytocin neurons in female rats are activated by somatosensory stimulation. These neurons drive the activation of magnocellular oxytocin neurons. The results of this study showed that parvocellular oxytocin neurons synapse onto magnocellular oxytocin neurons, causing oxytocin to promote communication between female rats. These results suggest that parvocellular oxytocin neurons translate mechanosensory information from the periphery into social behavior. The idea that somatosensory inputs selectively activate magnocellular oxytocin neurons was reinforced by the AirPuff data. Application of research findings. Clinical outcomes of schizophrenia, post-traumatic stress disorder, and autism spectrum disorders have been improved through intranasal oxytocin application. However, there is ongoing debate concerning the validity of these findings. Researchers suggest that inducing endogenous release of oxytocin could be a more reliable way to utilize the benefits of the neuropeptide. This study proposes a combination of gentle touch, intranasal oxytocin application, and/or social interaction could be an effective tool to treat various mental diseases in humans that are affected by a compromised oxytocin system. What could have been done differently? An experiment showing how interaction with multiple female rats could affect activation of oxytocin neurons. Does the interaction with multiple rats cause activation of more oxytocin neurons? Overall, the research methods used in this study were appropriate, and the researchers provided sufficient explanations of their methods and findings. Limitations. Only virgin female rats were used during this experiment. Since maternal experience can impact oxytocin neurons, it would be interesting to see how that could affect these experiments. Interactions between virgin females and males could have also provided interesting discoveries. Further research to understand the precise ascending pathways from cutaneous nerves via the parvocellular oxytocin to magnocellular oxytocin circuit to 4 brain regions controlling social behavior is still needed. This slide includes citations.